Hello everyone, this is Anna, a third-year immunology student. My dearest aunt, who felt constant discomfort in her breast, recently went to see the doctor. Unfortunately, she was diagnosed with breast cancer last week. All of my friends and my entire family were shocked by this bad news. Therefore, my classmate and I decided to apply our knowledge and do some further research on breast cancer immunology. The following are information from our immunology lectures and several scientific papers we found. We're going to present our findings to my aunt so that she can gain a comprehensive understanding on her illness and the following treatment. We can't cover cancer immunology without mentioning regulatory T cells. They are indeed a double-edged sword in our immune system. These are specialized T lymphocytes that normally protect us from autoimmune disorders, but can also suppress functionality of other lymphocytes upon recognition of tumor-associated antigen from cancer cells. As we know that FOXP3 can be induced by stimulating the receptor of CD4 positive and CD25 negative T cells, and functions as an important regulators for Treg cell development and function. A study which involves the participation of adult female patients with primary breast cancer who have just undergone breast surgery have shown an increase in FOXP3 mRNA expression in their breast cancer tissue compared with its expression in normal breast tissue taken from the site far from the tumorous lesion. Another previous study shows that the increased TGF-beta1 expression in breast cancer tissue will inhibit the generation of many effector T cells and antigen-presenting dendritic cells, thereby masking the adaptive immune system from recognizing tumor cells. The same study also concluded that the higher level of CCL22 in breast cancer tissue promote the communication between Treg and dendritic cell in the lymph node upon CCR4 binding. We are all familiar with natural killer cells as innate immune cells that play a fundamental and crucial role in cancer immunosurveillance and contribute to anti-tumor activities. The mere absence of MHC class I molecule can cause the activation of NK cells. Upregulation of stress-induced molecule like NKG2D ligands on tumor cells is a typical marker for NK cell killing. T cell infiltration is also enhanced by NK cell along with the cytokine production. The exact mechanism of how NK cells interact with breast cancer cells is not well understood yet. Nonetheless, several studies have shown that different subtypes of breast cancer cells show different expression of NK cell ligands, for example, inhibitory HLA members. In response to immunosurveillance, tumor cells have evolved different mechanisms to escape from the control of NK cells. Those invading mechanisms could be further studied and developed into potential anti-tumor treatment targets. One interesting study noticed an abnormal NK cell dysfunction in obese patients with breast cancer. Obesity interferes with NK cell function by affecting the NK cell metabolism and trafficking in the breast tumor microenvironment. Adipose tissue causes a reduced expression of receptors and ligands such as NKG2D and NKP30 in obese cancer patients. Also, NK cells have been shown to have the ability to absorb free fatty acid in circulation. NK cells with absorbed free fatty acid have a reduction in the ability to release cytokine, such as interferon gamma. Another crucial type of cell we would like to introduce here is the tissue-resident memory T cell. Studies demonstrate that one subpopulation of the tissue-resident memory T cell belonging to the CD8-positive tissue-infiltrating lymphocyte contributes to the peripheral immune surveillance and viral immunity in both human and mice with breast cancer. Patients with early-stage triple-negative breast cancers maintaining a higher level of tissue-resident memory T cells tend to have improved prognosis and live longer. Patients with advanced stage breast cancer with a larger population of tissue-resident memory T cells respond more actively to anti-PD-1 antibodies, which are a type of anti-tumor treatment. CD8-positive tissue-resident memory T cells also produce a large number of cytotoxic molecules like granzymes and immune checkpoint proteins, which are potential targets for immunotherapies as well. Intensive studies have elucidated that different immune cells interact with breast tumor cells differently. Some of them promote tumor invasion while others control its progression. However, further studies on investigating the detailed mechanism for these interactions will lay the groundwork for developing novel immunotherapy against breast cancer and other solid tumors. We hope our presentation can relieve your stress and anxiety, Andy. Please stay positive since there are so many effective treatments available. We sincerely hope that you will be restored to health.